Hello everyone, welcome to this presentation on nanomaterial research work titled as Filling of Carbon Nanotubes with Nanoactive Metal Species and Their Studies on the Physico-chemical Behavior. This research directly focuses on the unique synthesis techniques of CNTs and their mechanical modification for the physico-chemical as well as biophysical application. For your kind information, I am M.Phil, a chemical researcher, and the overall research work was a partial dissertation of the degree of Master of Philosophy in Chemistry from Bangladesh University of Engineering and Technology, shortly very renowned as BUET. Okay, let us come to the mainstream of this research work. First of all, I want to talk about nanomaterials. We know nanomaterials are the highly exclusive and active materials, and this is because of their smaller sizes. So they are very efficient than the other general sized particles. Among the nanoparticles, carbon nanotubes, shortly CNTs, are the high potent research area, and the authentic synthesis way and mechanical modification for the various physico-chemical and biophysical applications are the main interest of this research work. Now the question arises, what is CNT? CNT is actually a graphene sheet rolled up in a specific manner to form a homocircular cylindrical structure. CNTs are generally of two types: single-walled carbon nanotubes and multi-walled carbon nanotubes. The usual properties of CNTs are electrical conductivity, strength and elasticity, thermal conductivity and expansion, field emission, high aspect ratio, high absorbency, hardness, and kinetic properties. Applications of the functionalized CNTs can be discussed from two major sides. First one is biophysical or medicinal sides, and another is non-medicinal sides. Application of the medicinal or biophysical sides can again be classified into two classes. One is CNT-based therapeutics and another is biomedical detection and imaging. CNT-based therapeutics includes drug delivery, gene delivery, stem cell-related therapies, and thermal therapy. And biomedical detection and imaging covers photoluminescence imaging, Raman shift detection and imaging, and photoacoustic imaging. From non-medicinal sides, functionalized CNTs can be used in the preparation of conductive or reinforced plastics, energy storage, conductive adhesives and connectors, molecular electronics, structural composites, catalyst support, CNT ceramics, stain-resistant textiles, air, water and gas filtration, thermal materials, flat panel displays, and in transparent conductive coatings and CNT-based plastic packaging. Now let's know about the main objectives of this research work. The main objectives of this research work are to develop a unique method to prepare CNTs, to establish a suitable way for the filling of CNTs with nanoactive metal species like nickel oxide, nickel, manganese oxide, and manganese dioxide, to study the influence of the metal oxides on the physico-chemical behaviors of CNTs, to explore the efficiency of CNT in drug delivery, and to explore the absorption and antibacterial effectiveness of the metal oxides loaded CNTs. The common preparation techniques of CNTs are arc discharge method, laser ablation method, and chemical vapor deposition method. Now let's know about our synthesis method. We have prepared CNTs using organic bulk chemical method. The carbon sources are treated under forced in situ pyrolysis by the iron nanoparticles. The overall synthesis process is the combination of three consecutive steps. First, the carbon sources are treated with iron salt and benzalkonium chloride by continuous stirring for 24 hours. The aliphatic aromatic mixture of four organic compounds, benzene, metaxylene, hexanol, and octanol, are used as carbon sources. Here benzalkonium chloride is a cationic surfactant to stabilize the iron salt. Next, hydrogen hydrate are added and again run the 24 hours stirring to get the iron nanoparticles in the coagulated mixture. Then the overall coagulated compound were heated by 700 degrees Celsius, maintaining inert atmosphere in a tube furnace to get the final fluffy product of CNT. The preparation of other 4 nanoparticles are done using individual synthesis techniques. Nickel oxide nanoparticles were prepared using precipitation method. Here nickel chloride and nickel nitrate are used as nickel salts and urea as precipitating agent. Nickel nanoparticles are prepared by controlled reduction of nickel oxide nanoparticles. Manganese oxide are prepared by forced hydrolysis of manganese to acetate, and manganese dioxide are prepared by oxidation of manganese to acetate by potassium permanganate. Mechanical modification of the synthesized CNTs are done by controlled encapsulation of nanoparticles in the hollow space of them. Encapsulation of nickel oxide and nickel nanoparticles into CNTs are done by the boiling of dispersed nanoparticles with CNTs in 100 degrees Celsius. Manganese oxide and manganese dioxide loaded CNTs are guarded by the in situ incorporation of the CNTs during the preparation of these nanoparticles. After loading, every loaded CNTs are separated and washed for several times with water and ethanol. Now it was needed to characterize the synthesized nanoparticles. The overall characterization are done by the following ways. First one is scanning electron microscopic analysis to know the external morphology of the synthesized nanoparticles and the field matrices. Second one is energy dispersive X-ray analysis to get the elemental identification of the synthesized nanoparticles and the field matrices. Next, X-ray crystallographic analysis to check the crystallinity of the synthesized nanoparticles. And Fourier transform infrared spectroscopic analysis to check the unwanted functional groups in the sidewalls of the synthesized CNTs. EDX modified scanning electron microscope uses a focused beam of high energy electrons to generate a variety of signals at the surface of solid specimens. The signals that derive from electron sample interactions reveal information about the sample, including external morphology to confirm the size and shape of nanoparticles. Modern SAM generates data in digital formats which are highly portable. The SAM photographs are taken with an average magnification of 30,000 multiplication and 5 to 15 kilovolt at room temperature. The study of the SAM images show the clear size and shape of CNTs and other nanoparticles as well as the field matrices. Carbon nanotubes are clearly observed by the SAM image, whether EDX data confirms its elemental identity by almost 100%. The tube shape are also looking very uniform and diameter was found in nano level. The abundance and growth uniformity indicates the success of this authentic synthesis technique. SAM and EDX data also confirms the nano-level growth and elemental identity satisfactorily for nickel oxide nanoparticles. And then manganese dioxide nanoparticles are also confirmed by this authentic characterizing technique. Accordingly, manganese oxide are also confirmed. SAM and EDX data also give the proper idea of filling status of CNTs. The difference between the amount of incorporated nanoparticles and the amount of retained nanoparticles give us the percentage of amount of filling. Here we can see the SAM image and in corresponding EDX data for nickel oxide encapsulated CNTs. And these are for the manganese oxide loaded CNTs. These for manganese dioxide loaded CNTs. And this last slide is for nickel loaded CNTs. X-ray crystallographer is a tool for identifying the atomic and molecular structure of a crystal, in which the crystalline atoms cause a beam of incident X-rays to diffract into many specific directions. By measuring the angles and the intensities of these diffracted beams, a three-dimensional picture of the density of electrons within the crystal formed. From this electron density, the mean positions of these atoms in the crystal, as well as their chemical bonds, their disorder, and various information can be determined. Here, X-ray diffraction spectral images and corresponding data are provided for this presentation. Here we can see that the X-ray diffraction image for CNTs displays the clear sharp peak at 25.72 position of 2 theta, which clearly confirms the respective compound is CNT. This higher density of electrons at this position is almost identical for every CNT. The other content of XRD results match with the standard X-ray powder diffraction values of the respected molecules. Using this XRD data of these nanoparticles, the actual crystallite size and shape were determined using Scher's equation and Nelson-Rayleigh function. The sizes of the crystals are calculated and the values got for the corresponding crystals are excellent. From table, the actual crystallite diameter for nickel oxide nanoparticles and manganese oxide nanoparticles were found as 6.036 and 17.697 nanometer respectively. From the literature, it is mostly found that nickel oxide and manganese oxide crystals are cubic shaped, and we know the crystalline parameter for the cubic shape is A. Using the XRD parameter, the value of A are determined for nickel oxide and manganese oxide nanoparticles. Here are the A values for nickel oxide nanoparticles, which are 4.196, 3.630 and 2.567. Where for manganese oxide they are 13.44, 11.570 and 11.037. By plotting these A values against Nelson-Rayleigh values, A0 values were calculated. This graphical plot is for the determination of A0 value for nickel oxide nanocrystal. And this plot is for manganese oxide nanoparticles. These calculated A0 values are considered standard by literature review. Thus, it is confirmed that the nickel oxide and manganese oxide nanoparticles consist of cubic crystals. Fourier transform infrared spectroscopy, also known as FTIR analysis, is an analytical technique used to identify organic, polymeric, and in some cases inorganic materials. A material's absorbance of infrared light at different frequencies produces a unique identification based upon the frequencies at which the material absorbs infrared light and the intensity of those absorptions. The resulting spectra produce a unique molecular fingerprint which can be used to easily screen and scan samples for many different components. In this research work, FTIR analysis to check the unwanted functional groups in the side walls of the synthesis CNTs is present. FTIR spectra of synthesized CNTs shows a lot of significant peaks in between 1400 to 1650 per centimeter, which indicates the CC stretching present in CNTs in ring form. Peak in 2368.4 per centimeter, which might be OCO stretching for linear molecules of carbon dioxide due to unwanted oxidation of CNTs. Small peaks in 690 to 760 per centimeter are for carbon-bromine band stretching. Unwanted element bromine came from potassium bromide used for the FTIR experiments, and peaks present in 2850 to 2921 per centimeter for OH bending, which may came from unreacted alcohols used as carbon sources. The above information confirms that the percentage of uniformal growth of CNTs are very good. The adsorption-based applications of CNTs have received considerable attention in recent years. In our study, the activation of CNTs plays an important role in enhancing their maximum adsorption capacity in the case of encapsulation of the adsorbent particles in the hollow space of CNTs. Adsorption of methylene blue dyes on the surface of nickel oxide loaded CNTs represents an important and intensively studied phenomenon. Activation of CNTs under field conditions with nickel oxide nanoparticles are reported here. Now let's come to the adsorption study for individual CNTs. This table is for the determination of the adsorption at any time by CNTs. table shows the calculation of adsorptions of methylene blue dyes by CNTs at any time in microgram scale from 0 to 240 minutes at every 30-minute intervals. Plotting the values of QT against time, the adsorption at equilibrium state as well as equilibrium time of adsorption are calculated. Here, the values of equilibrium adsorption at any time is 56.01613 microgram per gram. Then, the rate of adsorption are checked out. From the figure, it is confirmed that the adsorption by CNTs follows the pseudo second-order kinetics. The plot shows a descending route which indicates that T by Qt decreases with time. From the graph, it is found that the rate constant K2 is 1.82 into 10 to the power minus 5 gram per microgram per minute using the linear form of the pseudo second-order kinetic of adsorption. At present, nanoparticles are the most widely used adsorbent with great success due to its large surface area, microporous structure, and a high adsorption capacity. For that reason, adsorption by nickel oxide nanoparticles are studied. Adsorption at any time Qt was calculated by using methylene blue dyes adsorbed in microgram scale and displayed in the table. The values of Qt against time are then plotted and QE, that is adsorption at equilibrium time, as well as the equilibrium time of adsorption are notified. Then the rate of adsorption was checked out. From the figure it is confirmed that the adsorption by nickel oxide nanoparticles also follows the pseudo second order kinetics. And in this case, the rate constant K2 is 1.27 into 10 to the power minus 4 gram per microgram per minute. The adsorption at any time was also calculated for the nickel oxide loaded CNTs. Then, following the same way, adsorption at equilibrium time as well as the equilibrium time of adsorption was calculated. Then the rate of adsorption was investigated, and in this case the rate constant K2 was found as 6.62 into 10 to the power minus 5 gram per microgram per minute. Now let's know about the overall findings from this adsorption study. From these adsorption studies, it was observed that the rate constant of CNTs and nickel oxides are 1.82 into 10 to the power minus 5 gram per microgram per minute and 1.27 into 10 to the power minus 4 gram per microgram per minute respectively. Here it should be clear that the adsorption capacity is higher for CNTs than nickel oxide nanoparticles, but it is comparatively time consuming. The rate constant of nickel oxide loaded CNTs are 6.62 into 10 to the power minus 5 gram per microgram per minute. The adsorption rate of nickel oxide filled CNTs are approximately 5 times higher than the individual CNTs. This increase of rate is because of the total increased surface area of the nickel oxide nanoparticles loaded CNTs. This excess surface area as well as the overall mechanical modification did impact on the rate of adsorption capacity. Now come to the antibacterial effectivity study as the biophysical application of CNT. We know antibacterials are among the most commonly used drugs. For example, 30% or more patients admitted to hospital are treated with one or more courses of antibacterials. The activity of synthesized manganese oxide nanoparticles was investigated against 7 bacterial microorganisms. Manganese oxide nanoparticles were synthesized by solution phase routes, and their interactions with these bacterial microorganisms are studied in broad spectrum. As yet, with all of the progress, many medications have unacceptable side effects due to the drug interacting with the parts of the body that are not the target of the drug. Side effects limit our ability to design optimal medications for many diseases such as cancer, neurodegenerative diseases, and infectious diseases. So, scientists now use nanotechnology to approach classical and novel drug delivery applications to maintain the targeting approach of the useful drugs. Reasons behind CNTs can be used efficiently as drug delivery materials. Carbon nanotubes have become strongest candidates mainly in the field of biomedical engineering, biotechnology and pharmaceutical nanotechnology after their discovery in 1991. These are because CNTs contain a significant hollow spaces that can be encapsulated with different desired chemicals as well as biological species. Carbon nanotubes have the potential to address the challenges of the combating infectious agents to achieve both targeted activity and combating infectious strains. This review focuses on the characterization, development and application of carbon nanotubes as nanocarrier-based delivery systems for achieving the desired drug delivery results in the different areas of infectious diseases. These are 7 bacterial species which are used for the overall studies of antibacterial susceptibility testing are Pseudomonas aeruginosa, Streptococcus pneumoniae, Vibrio cholerae, Escherichia coli, Salmonella typhi, Enterobacter cloci, and Acinetobacter baumannii. Antibacterial activity measurement of individual pure manganese oxide nanoparticles and manganese oxide nanoparticles loaded CNTs were done by Muller-Hinteregger disk diffusion susceptibility testing method according to NCCLS, that means National Committee for Clinical Laboratory Standards and International Guidelines. The overall techniques of preparing the dishes are observed here for the susceptibility testing. After 16 hours of incubation, each plate were examined and measured the diameters of zone of complete inhibition. The zone of inhibition for Escherichia coli and Pseudomonas aeruginosa by the pure manganese oxide nanoparticles are shown here. And here susceptibility testing for 4 bacterial microorganisms were done, which are Vibrio cholerae, Enterobacter cloci, Salmonella typhi and Acinetobacter baumannii. And this image shows the zone of inhibition for Streptococcus pneumoniae. Table shows antibacterial activity of manganese oxide nanoparticle suspension of 0.25 gram per liter against following pathogenic organisms. The diameter of zone of inhibition of different volumes for pure manganese oxide nanoparticles were compared with the standard ciprofloxacin dishes. Here we can see that pure manganese oxide shows very good result comparing with standard ciprofloxacin of same amount. Now let's observe the dishes for manganese oxide encapsulated CNTs using same bacterial strain. The left dish shows the zone of inhibition for Streptococcus pneumoniae, where the right one is for Salmonella typhi and Acinetobacter baumannii. And here we can see the zone of inhibition for Vibrio cholerae and Pseudomonas aeruginosa for the loaded CNTs. The diameter of zone of inhibition of different volumes for manganese oxide encapsulated CNTs were compared with pure manganese oxide nanoparticles. The results for this loaded status of manganese oxide nanoparticles are also very good and highly acceptable. Now let's know about the findings from the antibacterial effectivity testing. Manganese oxide encapsulated CNTs show very good results, and CNTs didn't resist the antibacterial effectivity of manganese oxide. The diameter of zone of inhibition is slightly less of about 1 to 3 mm, which is negligible comparing with the zone diameter of direct manganese oxide. As CNT is a well-established drug delivery agent, and from our antibacterial experiments, the effectivity of drugs are comparatively unchanged, so it can be said that the CNT can be easily used as a drug delivery agent on the issue of antibacterial treatment. Reviewing the current stage, we can see that the carbon arc discharge and laser methods are the techniques those produce a complex mixture of components and requires further purification. The uniformity in size and shape was rare present in these current methods, and it has been still unclear how to scale up production to the industrial level using these approaches. On the other hand, biochemical method of preparing CNTs is a new idea by which the integration of carbon nanotubes by this manufacturing process may solve the continuous growth with uniformities. Filling hollow carbon nanotubes with chosen materials opens new possibilities, which efforts to utilize the nanotubes' outstanding physical properties in the manufacture of composite materials, as well as in other practical applications. CNTs are relatively new adsorbents and hold interesting positions in carbon-based adsorptive materials. In this research work, adsorption by nickel oxide loaded CNTs provide the quick rates because of the greater surface area of the adsorbents, which enhance intraparticle diffusion. Researches have already been reported on CNT-based drug delivery. In this research study, the efficiency of killing activities were tested for manganese oxide encapsulated CNTs, which confirms that using CNT as drug delivery vehicle in the field of antibacterial treatment would be possible in near future. This overall research work focuses on the easy and smooth synthesis of CNTs, their proper encapsulation technique and functionalization, nanocomposite formation, enhanced physico-chemical behaviors, and drug delivery efficiency in terms of antibacterial treatment. The observed advanced surface active characteristics and drug delivery efficiency show promise for numerous other improvements in various advanced physico-chemical behaviors, biosensing, cancer cell demolition, DNA translocation, stem cell therapy, curing neurodegenerative diseases, and curing of infectious diseases. Whether the easy and uniform synthesis technique of CNT can be a big support for the all advanced level research work and cost-effective treatment for the mankind. I'm highly attracted to this advancement, and that's all of this presentation. From the higher intention on nanomaterial research work in physico-chemical and biophysical area, I got admitted in Bangladesh University of Engineering and Technology, that is BUET, on the session of April 2013 as an M.Phil researcher. BUET is the best science and engineering education-based university and generally gives the maximum facilities to the students and researchers at its range. To convey thus research work to the significant outcome, I have worked for almost 3 years mainly in the material science laboratory under the Department of Chemistry. In the meantime, I had to work in some other laboratories in and outside the university. One of them is Bangladesh Council for Scientific and Industrial Research, which is authorized as the main scientific research center by the government of Bangladesh. For the purpose of the inert pyrolysis and XRD characterization of the synthesized nanoparticles, I had to work in 2 different laboratories of this research center. ICT-DRB is another one, which is internationally renowned for microbiological research work. Here I had to work for more than 3 months in their bacteriology laboratory to accomplish highly intensive biophysical work of antibacterial susceptibility testing to confirm the drug delivery behavior of CNT. Working with nanomaterials was a big interest, and being awarded the higher degree of Master of Philosophy for the successful completion of this research work was the big achievement. It was the morning of 7th September of 2016 when I presented the overall research work at the Board of Examiners and got a big hand. Just 2 days before, it was also admired by the departmental professors, students and researchers after a seminar presentation. The matter of joy is 3 papers are just waiting to get published in the international journals. I'm grateful to my academic supervisor and to all other professors, scientists, lab mates, and lab assistants in and outside the university for their plentiful help and valuable suggestions. I'm grateful to my family members and to all well-wishers. Thanks everyone to have patience in observing this presentation.